Hey guys, just a quick update. In next Sunday's video, I'm going to be covering the 2018 annual Cure SMA conference. This conference was held in my hometown of Dallas, Texas, about a week and a half ago. And while I'm still waiting on some of the photographs to come back so that I can include them in the video itself, this video will be up on my YouTube channel next Sunday. Also, be watching my YouTube channel on Wednesday, because at the end of the conference, they held a Q&A session. And in this Q&A session were all of the researchers and the doctors from the various pharmaceutical companies. I was allowed to ask a question that's been on my mind for quite a while, and I think it's a question that you've had as well. And I think you'll be shocked at the answer that they gave me. So make sure that you watch out for the video on Wednesday. Okay, on with the show. Hey everybody, welcome to the SMA Journey 51 vlog. Okay guys, in this week's video, I'm going to be introducing everybody to a company by the name of Envisagenics and their new drug discovery platform that they call SpliceCore. SpliceCore is nothing more than a great big database of information pertaining to RNA splicing. RNA is an acronym that stands for ribonucleic acid, and what RNA is, in layman's terms, is that this is the in-between molecule that reads the data in DNA so that our bodies can produce a functional protein. If Envisagenics can successfully learn why this protein is not being developed at the level that it should be, then this may hold a key component for those of us with SMA so that our bodies can not only sustain the muscle that it's got, but also have the ability to build new muscle. Watch the PowerPoint presentation that I did, and afterwards I'll come back and I'll give you my final thoughts. Thank you. Who is Envisagenics? And what is SpliceCore? Envisagenics was founded in 2014 as a spin-off company from the Cold Spring Laboratory with the goal of developing treatments through RNA splicing analytics and artificial intelligence. Envisagenics announced that they've been given a 2-year, $1.5 million grant from the National Institutes of Health to further advance their new biomarker and drug discovery platform called SpliceCore. A Small Business Innovation Research Phase 2 grant will help Envisagenics in their efforts to further develop SpliceCore, a cloud-based drug discovery platform that uses machine learning and artificial intelligence to analyze ribonucleic acid sequencing data. Such data is important to the discovery of new RNA-targeted treatments. RNA is the in-between molecule in the process that reads the information in DNA to generate a functional protein. Errors in RNA splicing that affect protein development are a cause of many genetic diseases, including spinal muscular atrophy. Envisagenics was awarded a $225,000 Phase 1 SBIR grant in 2015 to develop an infrastructure capable of analyzing significant amounts of RNA sequencing data in a scalable manner. That first grant was also used to help build the largest database to date of RNA splicing events, and to develop machine learning algorithms to prioritize splicing errors relevant to diseases like SMA. The Phase 2 grant will allow the company to expand the knowledge base and predictive functions of its SpliceCore platform, which focuses on RNA splicing. Martin Ackerman, co-founder and chief technology officer of Envisagenics, said the following in a press release, quote, "The discovery of disease-causing proteins was at the center of pharma innovation for decades. But the new century brought us not only better knowledge of genetic information, but also the computer power to interpret it. RNA splicing treatments that we develop target the flow of genetic information so disease-causing proteins cannot be formed in the first place." End quote. Work supported by this SBIR Phase 2 grant will take place at Cold Spring Harbor Laboratory under the leadership of Dr. Adrian Craner, a specialist in RNA splicing, and at Rockefeller University under Dr. Thomas Tussell, professor and head of the Laboratory of RNA Molecular Biology. Maria Luisa Pineda, company co-founder and chief executive officer of Envisagenics, stated the following, quote, "We're excited for the next stage of growth for the company to leverage the power of artificial intelligence and RNA sequencing data internally and in collaboration with biopharma partners to unlock new treatments." End quote. In the last slide of my presentation, I would like to give everyone my opinions regarding Envisagenics' drug platform, SpliceCore. In earlier videos, I told everyone that Cytokinetics' drug reldesemtiv worked at the muscular level, and Biogen's drug Spinraza worked at the cellular level. Envisagenics' drug platform SpliceCore, if successful, could bridge the gap between Cytokinetics and Biogen. Biogen is working to get the SMN2 gene to either produce a full protein or to allow this gene to produce more protein than it's currently producing. In my opinion, if Envisagenics can successfully accomplish their goals and be able to correct the errors in RNA splicing, the 1-2 punch that Cytokinetics and Biogen had may become a three-way party. It seems as if every time we blink our eyes or turn around, new medical techniques and advanced procedures regarding medical testing are not only getting those of us with SMA excited, it's providing another glimmer of hope that all SMA patients can cling to. With all the pharmaceutical companies, clinical trials, and new drugs that are being brought to market, how can those of us with SMA not be excited about our futures? Okay, so I hope you enjoyed the PowerPoint presentation that I did for you, and I hope you got some information out of it that you can use as well. If you enjoyed this week's video, give me a thumbs up and subscribe to my YouTube channel. I would greatly appreciate it. Remember, when you subscribe to the channel, make sure that you click on the bell icon. That way you'll be notified of any new videos that I produce. I hope everybody's had a fantastic week. God bless you, and until next Sunday, bye-bye.